with us Dr. Troy Milley from the School of Medical Sciences. So Troy did a bachelor's in physical education from the University of Otago and did a PhD in exercise metabolism from the University of Melbourne. He did postdocs at Monash and ETH Zurich, where he was looking at the role of reactive oxygen species in glucose homeostasis and metabolism signaling pathways. So he then returned to New Zealand in 2016 as the recipient of a Rutherford Discovery Research Fellowship. And so his research continues to focus on molecular mechanisms underlying metabolic disease and how exercise can prevent the development of these conditions. So he's going to talk to us about some of the work he's been doing in this area.
Thank you. Thank you for the introduction and the invitation to come down and talk today. As Ned said, I'm interested, as a way of introduction, I'm sort of interested in health, exercise and metabolic disease, and trying to understand how we go from a state of health to one of insulin resistance, type 2 diabetes and other metabolic disease, and how exercise can stop that. Today I'm going to speak largely about metabolic disease signaling and touch briefly on the exercise and other things towards the end, where I'll spend probably half an hour talking about data we've published or in the process of publishing that largely comes from postdoc work and that we're sort of following up here, and then talk very briefly about some new sort of areas that might be more interested for this crowd. And firstly, apologise because there's a heap of cellular signalling in here, so I hope you can bear through it. So interest is in obesity and type 2 diabetes. This figure shows the change in BMI, which is kind of a pseudo measure for obesity from, was it late 1980s to 2014. This is in America, and we can see this shift in the curve upwards with the increased rates of obesity. But one thing to point out is that we've got the curve, and it's also not just shifting itself up, it's changing shape as it goes up. So that to me indicates that it's not one factor causing this change, and there's lots of underlying factors. So it's not such a simple disease, and there's many things going on. This is why we need to spend time understanding it. And kind of the emphasis we have now on exercising more and eating less, while it's good in theory, it's clearly not working because this is still going up. So we've got to think of new approaches. This is why it's important. Obesity, type 2 diabetes, metabolic disease causes lots of issues. Central nervous system issues. We're going to focus today largely on diabetes and liver disease and insulin resistance. But there's a host of factors that diseases that are upregulated by obesity, and not everyone gets these diseases. So it's really interplay of what's happening and trying to understand this. One sort of underlying factor around a lot of these diseases is inflammation and reactive oxygen species. So reactive oxygen species are produced by the mitochondria and from other places like the NADPH oxidases. And these have been historically implemented primarily in disease, muscle wasting, cancers, and they occur as a normal byproduct of oxidative metabolism. However, more recently, what we see is antioxidant treatments to try and to stop these free radicals and reactive oxygen species as a way of slowing disease progression and the development. However, what we're seeing in the literature now is this really isn't working. And in most cases, in some mouse models of obesity and type 2 diabetes, we see some effect with genetic modification, but overall they're not having much of an effect, and in some cases they're actually promoting disease. Particularly, I think it was this bowel cancer that's coming up. High doses a long time with vitamin C has had an increased risk. So this suggests there's something a lot more complex going on, and probably makes sense that it's not just as simple as we have reactive oxygen species, we block them, we stop sickness. And more in the last decade, it's come to light that reactive oxygen species plays a role in physiological responses and normal cellular signaling. So acute responses such as improving insulin sensitivity, initiating growth factors, immune responses. And these also lead to adaptive stress responses, and there's growing evidence that oxidative stress caused by reactive oxygen species that happens during exercise or as an early sort of stress can promote healthy aging and exercise adaptation. So there's a lot more going on. The idea is potentially these antioxidants might be blocking some of this and preventing these positive adaptations and therefore promoting disease. So this figure here is a very basic overview of the oxidative stress and reactive oxygen species signaling. We've got the parental ROS, superoxide. This is broken down by superoxide dismutase to hydrogen peroxide, which then can be broken down further by catalase to water and oxygen, or by the glutathione peroxidases to water. So in the lab, in Tony Tagana's lab in Monash, before I got there, what they did, they brought mice that don't have GDX, the enzyme GDX. So they can't convert this hydrogen peroxide to water through this pathway. So in theory, they have an increased levels of hydrogen peroxide. We're interested in type 2 diabetes and insulin resistance. So this is the kind of figure you're going to see a few times through this talk. This is blood glucose, and this is time. And what we've done here is injected the mice with insulin, and we followed the decrease in blood glucose levels over this time. The further the increase in blood glucose, decrease in blood glucose levels, the more insulin sensitive the mouse is. So we put these mice on a high-fat diet that initiated like a pre-diabetic state. So they put on a little bit of weight, their blood glucose as well weren't overly increased. And then we compared that to mice that don't have GDX, this antioxidant, and found that there is an improvement in insulin sensitivity. So this suggests that in this pre-diabetic state, ROS are actually promoting insulin sensitivity, which is kind of opposite to the dogma where reactive oxygen species promote disease. So just to take a step back from that to go into provide an overview in insulin signaling, this is what happens when we have a meal. Glucose goes into our bloodstream, and we need to get it out of our bloodstream into our tissues for it to be used. However, it doesn't freely transport at a high enough rate to lower our blood glucose levels. So we have pancreas that releases insulin into the bloodstream and it travels to our peripheral tissues, such as the liver and the fat and the muscle. It binds to the insulin receptor and allows for the glucose to move to the cell surface and facilitate the transport of glucose into the cells, and therefore lowering our blood glucose levels. It also acts on the liver to stop glucose being produced by the liver and to stop the further rise in our blood glucose levels. In type 2 diabetes, we see this is stopped through mechanisms we're still trying to understand. So insulin fails to stop the release of glucose from the liver, also has a less effect of stimulating glucose transport up into the tissue. So now if we zoom in on the muscle, we'll go through this. Reactive oxygen species have been sort of implicated in causing damage to the pancreas to stop a decent level of insulin being secreted, causing damage to the liver and the muscle to stop it reacting to insulin. So this is kind of inverse to what we were seeing in the previous figure. So now if we zoom in on the muscle itself, we look a little bit more at the signaling pathway. We have insulin that's binding to the insulin receptor, and through a cascade of rather simplified complexes here, we see insulin acting with the insulin receptor substrate and sending a signal down to the glucose transporters to move to the cell membrane and transport glucose into the cell. And this process becomes blocked with type 2 diabetes, and we can't get the glucose into the cell. However, what I haven't put on there initially is there are other things acting on this pathway to also stop it. These are called protein tyrase and phosphatases. They stop signaling, they turn off, they take the phosphate group off these and effectively stop the signaling from happening. They're like a rheostat to stop the signaling happening after the signal's gone. These phosphatases are susceptible to oxidation by reactive oxygen species. So these reactive oxygen species can turn off these phosphatases and promote signaling. So the idea in the earlier figure I showed is that by knocking out the antioxidant enzyme, we're promoting this signaling of this cascade to enhance insulin sensitivity and improve insulin sensitivity. So the idea there, where I sort of picked up on this story as well, what about conditions of severe obesity? So we saw this enhanced insulin sensitivity in mice that don't have this antioxidant under conditions of pre-diabetes. Perhaps under severe obese conditions, these reactive oxygen species are much worse, and they're going to make their insulin resistance in type 2 diabetes even worse. So what we did, we compared these GPX knockout mice on diets that promote just a low-fat normal chow diet, a pre-diabetic diet, as we already shown they have increased insulin sensitivity under, or a diet that induces severe obesity. So we can see this is the percent body fat. With an obesogenic diet, it's up around 40% of body weight, whereas a normal chow-fed mice is around 20%. And this obesogenic diet actually elevated blood glucose levels and increased oxidative stress. So GSH-GSS-G ratio is a measure of oxidative stress. If you decrease this value, as on the right-hand side of the figure here, it shows you systemic oxidative stress in the blood.
Or is the obesogenicity due both to overload with sugars and fats?
Yes, so it's got a high sucrose in it, as well as about 23% fat compared to about 5%.
Kentucky Fried diet.
It's basically a Western diet, but it's easy to explain as you say a high-fat diet. Well, this one here has actually got a bit more fat in it, but a lower sucrose. This is sort of the two we've used previously.
Is fat mainly just a problem if you eat meat?
No, it's a problem if you eat other. There's a lot of deep fried stuff. Things like that, you're a lot of energy dense. So I think it's a global approach rather than just targeting one. But so the main point here was we're seeing this oxidative stress in this diet with obesity. So we thought the systemic oxidative stress increase in the mice, they're going to be less glucose tolerant. But what we actually saw was no difference between the two mice when they were on this really high-fat diet. And we can see that in the insulin tolerance test, where we give them insulin. We see a drop in their blood glucose, and this is similar between the two mice. And the alternative, if we give the mice glucose, this increases their blood glucose levels, and it's how quickly they can clear this. And we see maybe a slight increase in the GPX knockout mice, but overall relatively similar. So we're quite surprised by that.
Expect the glucose tolerance test and the ITT to be much the same?
I think the glucose tolerance test focuses more on the liver, so you quite often get differentiate between them. They're both rather crude measures of glucose sensitivity, but they normally match, but not always. And I think for my experience, the GTT is more of a measure of the ability to stop glucose being released from the liver, but it also has a bit of both in it. So this was quite surprising to us. It didn't exacerbate the insulin resistance. So we did a few more measures, and what came out was that these mice actually had lower insulin levels. So that didn't really make a lot of sense either, because we saw similar GTT. And GTT, glucose tolerance, is driven by insulin. So if we had lower insulin levels, how come they have similar insulin sensitivity or glucose tolerance? So we delved into that. This is just from a fed and a fasted blood glucose levels of the mice. We delved a bit further, and we did what's called an intravenous glucose tolerance test, where we can infuse glucose and take samples very quickly, so we can get like 2-minute samples after we infuse glucose to look at the secretion of insulin. And we can see here that the GPX knockout with this insulin secretion assay have a lot lower insulin secretion. So to kind of summarise, we looked at a few different signalling pathways, and we found that PDX1, which is a regulator of beta cells, we think through oxidation of phosphatases, was reduced. But that's sort of not the kind of story focusing on today. We wanted to understand how this insulin signaling or changes in insulin signaling might be driving the obese phenotype. So we moved from that, and we'll come around here while we focused on the liver. But to give a little summary of what happens in the liver during obesity is that we've talked about insulin signaling, glucose uptake, glucose lipogenesis. So insulin promotes lipogenesis, glycogen synthesis. That does a whole lot of other host factors, but to simplify it down, I'm just focusing on the metabolism ones here. It also turns off the making of glucose from non-glucose sources and the breakdown of lipids. When we become insulin sensitive, insulin resistance in our liver, this isn't all targeted equally. And we can see suppression during selective insulin resistance of these pathways, the glucose uptake pathway, the lipogenesis, lipolysis pathway. But the lipogenesis pathway kind of conversely seems to be upregulated. And this is a little bit of a conundrum in the research, is how we can see a similar insulin signaling pathway having divergence effects. Some aspects being increased and some aspects are being decreased. So this is sort of got us more interested in this area, and we started to focus a little bit on the liver. And this is what happens in insulin resistance, largely leads to a non-alcoholic fatty liver disease, where fat storage through lipogenesis is highly upregulated, and this has been estimated in obesity cases and insulin-resistant cases. Up to 80–90% of people have some degree of non-alcoholic fatty liver disease. So it's a massive problem. The normal hypothesis around this obesity is that, and its development of type 2 diabetes, is that we have an energy intake greater than energy expenditure. This leads to obesity. We get insulin resistance, our insulin doesn't work, and as a default we get a hyperinsulin secretion. This is to try and rectify this insulin resistance by producing more, and this leads to type 2 diabetes. However, there's more evidence gaining that hyper-insulinemia might happen earlier than expected and might actually drive some of this phenotype. In particular, these guys showed a few years ago that we genetically modify mice so they can't produce enough insulin under these high-fat conditions, and they're protected from high-fat diet-induced metabolic syndrome. So that figure up there is just to highlight that the GPX1 knockout mice we're talking about and the wild-type mice had similar glucose tolerance. However, they're producing less insulin, so they must be more insulin sensitive somewhere, because they're still able to reduce the level of glucose in their blood even though they've got low insulin. And now if we have a look at more of cellular signaling to look at the activation of AMPK, AKT, which is the downstream of the insulin receptor. And we can see each of these blocks here is an individual mouse. These are mice treated just with saline, and these are mice treated with insulin. And we can see a nice increase here, which is a lot less in the wild-type mice. So despite having less insulin, these guys and more ROS, they're actually more insulin sensitive in the liver. So how does this then affecting the liver, that these guys are actually more insulin sensitive in the liver? So we looked at plasma markers of liver damage, ALT and AST, and we can see the GPX knockout mice on the right-hand side of each of these figures have a lot lower levels, so they're having less liver damage. And then if we look at the histology here, it's quite clear that all these white dots in the GPX wild-type mice is fat, and when they're knocked out, we get less fat storage in there, and the bottom figure is just more of a lipid marker. So these guys are more insulin sensitive in the liver, and they seem to be somewhat protected from fatty liver disease. So the question we asked there was, is this effect of knocking out the GPX itself, or is it an effect of the reduced insulin? And we were sort of leaning towards the reduced insulin levels that's driving this. So we generated mice that don't have GPX specifically in their liver. So the first mice I've been talking about don't have GPX anywhere in their body, and these are liver-specific GPX knockout mice. So this is a Western blot of hepatocytes taken from these mice, grown in a dish. We can see the GPX levels, they've got nothing there compared to the wild type, or this lox mice. So, long story short, we put these guys on a high-fat diet and had a look at the lipids in their liver, and we found that they got similar levels of lipids or fat in their liver as the wild-type mice. So this indicated that this is an effect that's independent of the GPX1 knockout. It's probably driven by the liver. But interestingly, and a story for another day, is that while these guys had similar levels of fat accumulating in their liver, they had a lot less inflammatory markers and markers of steatosis happening in the liver. So perhaps this knocking out of the antioxidant is pushing some protective effect from liver disease. And we went on and challenged these mice with other models of liver disease and found they actually protected to some extent from the fibrosis and steatosis of other conditions. But for today's story, we're talking about the insulin receptor. So this to us indicated it's probably insulin driving. So now we move to the ETH in Zurich, where we developed another model of mice looking at whether we can reduce the expression of the insulin receptor and how does this affect fatty liver disease and metabolic syndrome. So these mice are quite a cool model. So most genetically modified mice, you knock out the protein, they're born without the protein, they live without the protein. So this is an inducible knockout system. So these guys are born normally, they have a Cree that's inactive, and then we can activate this Cree by giving them a bit of tamoxifen when they're adults, and this knocks out the protein once they're already adults, so you negate any developmental effects that might be happening. So we can see here in the liver and fat, so this Cree we used targets all peripheral tissue. We don't get any central effects in the brain. But it's pretty effective. We can see in the liver with the peripheral insulin receptor knockout mice have no insulin receptor in their liver. So we just had a look at these mice, and we see the mice there without the insulin receptor, as you expect, insulin resistant, because they have no insulin receptor. You see really high levels of fed blood glucose and really high levels of insulin. So that doesn't really help our cause, because we're trying to study insulin resistance, and these guys are already insulin resistant, as you expect. So for the rest of this talk, I'm going to focus pretty much solely on the peripheral partial insulin receptor knockout mice. So these are heterozygous mice that have about a 50% reduction in the insulin receptors, we can see here, compared to the mice that have all the insulin receptor gone. So what we did was fed these guys for 15 weeks till they're adults on a chow diet, treated them with tamoxifen, and then put them on a high-fat diet for 10 weeks to look at their metabolism. What we did is, this is very similar to a GTT, but it's a little bit more sensitive. We found no differences in their GTT, but we did a fast refeed experiment where we have the mice fasted overnight and then feed them the next morning and follow their blood glucose rise, and then fast them again and refeed them again. And what you can see is the fold change, the decrease in blood glucose levels, decreased more in the peripheral knockout mice, and we did what's called a pyruvate tolerance test. So this is a test that specifically looks at liver function. If we give the mice pyruvate, you get gluconeogenesis, production of glucose in there from their liver, and this increases blood glucose levels. And the quicker you can turn that off, is under high fat conditions indicates how your liver is functioning better. So these peripheral knockout mice had improved liver function through this somewhat crude test. And then we went on to have a look at the histology and the blood markers of liver damage. And again, we saw plasma markers of liver damage substantially reduced by reducing the level of the insulin receptor. And we looked at the H&E staining to look at fat, and we see the peripheral insulin receptor knockout mice will reduce levels of the insulin receptor. Pretty much completely protected these mice from fatty liver disease, without having massive consequences on their ability to regulate blood glucose levels. So it's quite interesting. It suggests that maybe our insulin receptor, we've got so much there, we only need a partial activation for it to regulate glucose homeostasis, but further activation might cause other effects, maybe on lipogenesis more. So the next question was is how does this partial peripheral insulin receptor ablation improve liver function during these conditions of metabolic stress? After measuring a number of signaling pathways, one we came to was measure the ADP to ATP ratio. So this is a measure of energy deficit. So you can see these guys have a higher ratio indicating they're in energy debt in the liver. And one of the known kinases to activate it in response to energy defect is AMPK. AMPK regulates a number of processes, some of which are involved in metabolism, being fatty acid oxidation, lipogenesis, gluconeogenesis. So this sort of fitted in with our hypothesis that we're getting increased fatty acid oxidation and reduced lipogenesis in these mice because that we could visually see less fat in their liver. So we plotted to see if AMPK was upregulated, and we see quite a clear phosphorylation as a measure of upregulation in the liver of these mice on the right. And some of the downstream markers like PGC1 alpha were highly upregulated as well. So PGC1 alpha is a master regulator of mitochondrial biogenesis. So it kind of fits with the idea as well, is that we're getting activation of AMPK, upregulation of mitochondrial biogenesis to increase fatty acid oxidation. And indeed we saw quite a robust increase in citrate synthase activity in these livers, which is a marker of mitochondrial volume. So we think these mice have more mitochondria, more active mitochondria, and they're able to use more lipid, and therefore they are not getting fatty liver disease. So this is the working hypothesis around this, and that we're getting reduced insulin receptor, we're getting less glycolysis as a result of less glucose being used, and then we're getting less byproducts of the TCA cycle, less free fat accolase, and this is getting less lipid storage. As a result, we're getting activation of AMPK. And as another sort of side story, we see changes in adiponectin levels. So we don't know if this effect is specifically liver. It could be the fat or other tissues feeding back to the liver to cause an energy deficit or to activate AMPK. So we can't really rule that out now because this is a global model. We think the AMPK is activating mitochondrial biogenesis and lipid metabolism markers. And as a result, we're getting more lipolysis, more use of the beta oxidation, and less lipid storage. So that's sort of where we're at with that story. And another way to kind of look at this data is through the signaling mechanisms. This is sort of in the last year or two has kind of come out as sort of where the insulin signaling pathway might diverge into two different areas for lipogenesis and gluconeogenesis. And that with selective insulin resistance, we get a downregulation of the FOXO pathway that regulates the suppression of glucose production. So it suppresses glucose production so we don't have chronically high blood glucose levels that then go on to cause coronary artery disease and many other pathologies. But for some reason we're getting an upregulation of the mTOR pathway. And this seems to be promoting lipogenesis and and inhibiting mitochondrial ability to adapt to this increased stress. So what we think was happening here is we're getting this negative energy balance by reducing the insulin receptor levels, or if we go back to our first model, by reducing the insulin levels overall. And this can sort of suppress this pathway, and we're still getting some small phenotype with the glucose signaling, but overall it's improved because the liver fatty acid is reduced. So this signaling pathway is very similar to that, what we see is associated with aging and longevity, and the strongest stimulus to promote healthy aging is calorie restriction. So one of the concepts around how calorie restriction improves our aging is that it reduces insulin and sort of brother molecule to IGF-1 signaling. So we can block this pathway. This leads to positive adaptations. Now here, usually associated with antioxidant responses, mitochondrial biogenesis, and the use of fatty acids and lipids more as an energy source. and activation of AMPK to promote adaptation, and leads to long living. And some of the clear evidence of this comes from monkeys. The one on the left side here, they're the same age, but the one on the right had about 30% less calories than the one on the right throughout at least the last few years of their lives. And you can see it's promoting a lot healthier phenotype. So we asked the question with these mice, are they going to be long-lived? Are they potentially going to age healthier? So this comes from the idea that, at least in C. elegans and Drosophila, it's pretty much unanimous. You can block almost every stage of the insulin IGF-1 signaling pathway, and these guys will live longer. In some cases, up to 50% longer than they would live otherwise. So the mean lifespan for a Drosophila is about 15 days. The maximum is around 25, and these guys can live up to 60 days with these mutations. It's not quite so clear in mammals that targeting just the insulin receptor or just the IGF-1 receptor. Some models say they do, some models say they don't. For example, if we heterozygous knockout of the IGF-1 receptor all over the body, the mice live longer. Tissue-specific deletion in the fat, which has turned out to be not so specific and it's supposed to be hitting the brain as well, they live longer, but other models don't. So we asked the question, can our model that reduces insulin receptor only once they're adult and only in peripheral tissues extend lifespan? So a couple of short slides before we go into the lifespan models is that these guys show adaptations that we think would extend lifespan and make for a healthy model. They show a slight reduction in fat mass, and we can see an increase in small adipocytes compared to a reduction in large adipocytes. And this is sort of frequently reported in mouse models that are long-lived. We can see actually under these chow-fed conditions, they're actually slightly more insulin sensitive in the liver, so it kind of fits with our high-fat diet data. There's kind of a debate on whether this is a good or a bad thing, because there's an idea that a lot of the models that are long-lived are slightly insulin resistant. We also looked at the mitochondrial biogenesis, and we see a trend for an increase in mitochondrial volume. It's measured by the mitochondrial electron transport chain here, plots, and citrate synthase activity. Thing is, we didn't see anything. We see the vehicle. These guys have just had the tamoxifen. They've just had the vehicle, not the tamoxifen, and this is sort of a control to show that our adult-induced ones are the same if we don't induce the knockout. We see their lifespan very similar. We see the peripheral insulin receptor complete knockout, so no insulin receptor expressed. As you may expect, they live shorter because they have very insulin resistance. I showed an early one. We were quite surprised our heterozygous mice that have partial reduction pretty much the same as the wild type. So that's disappointing after a few years study. So it begs the question whether targeting insulin receptor is worth it in mammals. The second question is, there's some evidence gaining that it's got to happen in the central nervous system, and this regulates healthy adaptations and aging. So this model, we're only affected peripheral tissue. And the third one was, do you have to restrict for targeting the insulin receptor during development for all these adaptations to take place to live longer, because these are adult-induced mice with a reduced insulin receptor expression. We don't know the answer to these. They're ongoing questions, so it'd be interesting just to target the brain or just to target during development earlier stage. So calorie restriction has been shown to have effects even in old age. So just to summarise, this is a data point of the talk, then I'm just going to do another 5 minutes showing sort of some more recent studies we're starting to work on away from mice as well. So as well as following up on these ones. So on the first slide, I tried to show that oxidative stress during metabolic overload does not further increase impaired glucose homeostasis and can reduce insulin sensitivity. So I'm fairly certain there's instances where reactive oxygen species do cause disease and they're very bad, but there's also conditions where they might actually be positive as well. Insulin signaling contributes to the development of metabolic disease. and that partial peripheral insulin receptor expression reduction in its expression doesn't extend lifespan in our model anyway. So, as I said in the first couple of slides, we're interested in understanding metabolic disease and obesity. So we've got energy in, energy out. Simple, right? When we break it down, it's not that simple. There's a lot of other things regulating our energy in, energy out, from the quality and quantity of the food we eat, what sort of governs that, how hungry we feel, how full we feel, what we preference, how much we absorb it. We don't have any control of this. So it's kind of, you've got to restrain yourself. So how much is choice not to eat more and how much isn't is sort of debatable. And our energy expenditure, how much we use when we're just sitting there, how much we use when we get cold and warm, how effective we are at moving, and our activity levels. So really, very few of these are under our conscious control all the time or easily controlled. And this has come with the idea that the genetic component is estimated to be 40 to 70%. So we're interested in unraveling what these pathways are and how they work. One study we're looking at is understanding our resting metabolic rate a little bit more. Resting metabolic rate is, in this figure, it's probably around 75, probably 60 to 75% of our energy expenditure. So physical activity on top of that is 20% max. So most of our energy is being used just sitting there, our cells just living. So if we can find out ways how this works and how this differs in people, and what we can do to promote this is a better weight loss or metabolic control pathway than just exercise. So one factor that contributes to a resting metabolic rate is activity of our tissues, and one tissue that's really active is our brown fat. This is sort of a newish realization that adult humans express brown fat, and this brown fat is full of mitochondria and produces lots of energy because it uncouples, and its purpose is to produce heat, not to store energy like white fat. This makes it really, we're sort of embarking on a little bit of a study where we try to measure brown fat. So this is the more accurate way, I'd say, to measure brown fat, and how it was recognised is that people are exposed to cold and a PET scan with a radioactive tracer to look at where the glucose goes up. We can see these areas around here and here, probably brown fat, and biopsies have shown this is brown fat, and this is where it's taken up. Doing this approach is not very cheap, and not always that ethical, particularly if you want to do children research and in healthy people, because you have to put a radioactive tracer in. So there's a new sort of method being developed. This is around using a thermal camera. The idea there is you're sitting there stable, and as you're cooled, your brown fat becomes activated and it heats up to heat up other parts of your body. So we're kind of using this theory to go around to measure that. And the idea is to kind of get an idea of how these brown fat levels are different in the New Zealand population, particularly Pacific, and Maori students at school, and how this changes. So this is some preliminary data we've been playing around with. This is a 5-minute cooling stimulus. One of our guys, you can see these areas here increase, and we can measure the distance and how much they increase in temperature, and we can see nice increase there. So we're sort of working with schools to try and measure our children's resting metabolic rate, their brown fat levels, and fat mass through a DEXA scan. The other area of interest we're really interested in is how exercise. We know exercise improves health. It does a lot of things. We're trying to understand how exercise improves health. It improves our insulin secretion, improves central nervous system function, cardiovascular function, changes our fat mass, and changes what we release from our fat cells. And there's a heap of complex pathways going on. So this is a little bit of data, is that we've got RNA sequencing from mice that have been with exercise, and more than 600 genes, mRNAs, change with this is 4 hours following exercise. There's a massive effect there. And to come back a little bit to the reactive oxygen species story, is that if we treat them with the antioxidant, only half of those genes are still changing. So this indicates that there's stresses that result from exercise that can be blocked, that can potentially be targeted to improve exercise-like adaptations. And one of the models we're working on at the moment is we can't knock out every gene very easily in mouse models and develop them every time or in humans. So we've got this model going where we can hopefully exercise cells and target these pathways. And you see, if you look closely, these guys are being stimulated by this stimulator up here, and then they're contracting away. And then we can take muscle biopsies from humans, which we do relatively frequently at the moment, and drive these cells and use people with different genetic backgrounds and study the insulin signaling, the exercise signaling molecules under these conditions. So that was my last slide. Just the thank yous now to people. This work's been done all over the place. This reactive oxygen species, GTEC stuff is done in Tony Takana's lab at Monash University, and with the help of some people from Melbourne University, Sof and Barbara. And then I moved to ETH in Zurich, where I worked under Michael Ristow and the Ristow lab. So there's several people, particularly Kim and Beata, for helping me out with studies there, and also Johannes, so it's Tony's lab, Michael's lab, and now a few people I'm working with, Peter and Lindsay Plank here at Auckland University, and Shannon's research assistant who's done a bit of the contracting cell stuff. And the funders, Novartis, NHMRC, European Foundation of Study of Diabetes for overseas work, and more recently some guys back here who are supporting my work back here. Thank you guys for your time and your attention.
Thank you, Troy. We have time for questions. Jill?
Thanks, Troy. That was really good. Really interesting models. As you know, I'm interested in bone development, bone regeneration, but particularly bone and aging and how it changes, and bone changes dramatically in diabetes as well. But it's not exactly understood how the diabetic metabolism is affecting bone. Obesity is normally reasonably healthy for bone, but as soon as we become diabetic, the microstructure of the bone changes and more commonly fracturing. And I'm just wondering with some of your models there, when you're sort of working between obesity and diabetes, whether we should be doing any bone studies that could knock one out and
we did do on the GPX knockouts there we had the DEXA scanner at Monash. We didn't see any massive differences there. But there certainly doesn't mean there's no difference. It's just the density's different. We haven't done a lot, and I can't comment too much on bone
it's
stuff.
It's more than just bone density. You've actually got to do micro CT to get the micro.
They definitely. Because
the densities can be similar, but they fracture a lot more than diabetic.
I haven't done too much work on the bone, so I can't comment too much on that at this stage.
By the way, the fixing this is fixed. It's
it is.
back.
Good. I want it in a couple of weeks. Holding on.
Of course, that relies on having an animal house in which you can get the animal stat in the first place, of course. But
well done, Peter.
The animal house questions aside, anything that raises AMP kinase in the liver should have some of the same effect. Metformin being an obvious example. But the other way to really get your AMP kinase levels up really, really quickly in the liver is with fructose. And what part in this process is that playing, especially given any diet you've got has got fructose in it?
It could be playing a role, but we're comparing apples with apples. These more wild-type mice and knockout mice are on the same diet, so we're seeing a further increase if AMPK is up under these conditions. We're seeing a further increase with knocking out the insulin receptor. So even though there's fructose on the diet, this is going to be an additive effect.
I'm just thinking if you ran them on a diet with no fructose, just a pure starch diet, and compared that.
Actually, what I did do, it's been a long time ago, we did it on a high sugar diet, so that's got heavy fructose in it,
Yeah.
without the fat, and we saw no differences basically in the phenotypes. And they really didn't put on any weight either in these conditions.
There might be a big driver for fructose in there.
So it might be more of an interaction with the lipids there in diet and the fructose to get these cardiometabolic effects. Maybe on one on its own is not the worst, at least in mouse models.
Or the paleo diet.
With a little bit of sugar on top. It's dead.
Troy, do you think the data that you're getting is good enough now to be applicable to a modelling study? I mean, are you working with anyone that's mathematically modelling the mechanisms?
I have no background or knowledge really in modelling and what would we really require to model, but I would think, I mean, these are kind of run-of-the-mill studies. A lot of people do these high-fat diets and look at signalling mechanisms. So if it's not enough in this study, I imagine it's out there. This is kind of a tried and true methods to understand metabolism. We age mice, we put them on a high-fat diet, we do glucose tolerance tests and tolerance tests, and it's run through in many labs around the world. So in that case, it should be.
So how much interest does that community have in modelling?
I have seen very little in it on the way of modelling, and I'm not sure
It's
what.
like the bone area, they need it.
I don't.
And so if it's like the bone area, they'd actually need it.
Yeah, I imagine a lot of people are in the same position as me, as I don't understand enough about how modelling works and how it can be advantageous to my work to delve into it.
But I suppose the other way of phrasing the question is, do you think that you're getting to the point where the system is too complex, given all the mechanisms going on, for you to really quantitatively understand what's happening without the use of models?
Yes, so that's a really good point, and I think the use of more bioinformatic and large data inputs now with proteomics and RNA-seq and putting these approaches together is probably helping us, and it would probably help perfect a model area where you can kind of put in all this sort of data and the stuff David James is doing now with the phosphokines, and where it's really large data sets, and there's going to need to come up with a way you can actually synthesize all those different inputs into one thing, rather than doing what we're doing, is rather simply knocking out one protein and see what it does and try and understand
You're
from there.
not frightened off by about 1,000 different genes affecting things, Peter? Or does that just make the problem that much more satisfying?
I suppose that's why I started the question off with the quality of the data. You know, whether the data is up to the task of being able to adequately drive more.
I think for simple questions, but they would have to be quite controlled, because as I said, everyone's doing this diet model and stuff like that, but people do different, might start in a slightly different age, slightly different diet. So I think if we can generate data that's really controlled, like a few groups where we do a high fat versus low fat under the same conditions, same temperature, same age, and then we can pull data from metabolic, from RNA-seq, then we can think probably more about modelling in that space. And I'd be quite interested in that for particularly our exercise stuff, because we can kind of do that now. We're doing that from a range from humans through to mice, through to cells, and that.
Maybe we should get a group of people together to explore that a bit more.
Yeah, I think that would be a good idea.
So one of the questions that I had was, so you talked about the peripheral knockouts for the insulin receptor. So, and you talked about the knockouts in the liver, but what happens to the receptor levels in muscle or in other tissues?
Yeah, so peripheral tissues, we did these in all the knockouts. We tested pretty much all the tissues, and the only place we don't get is in the central nervous system. The hypothalamus, we don't even in the maturity, we don't get much of a knockout. And I think it's only a partial knockout of brown fat, but the rest is, as you see on those plots. I think it's more not, the Cree's still there, but you're not getting as good recombination with the tamoxifen doesn't get into the central nervous system quite as well as the other tissues. So it makes it an interesting model. You could do the other way, is do an ICV injection into the brain and just have it knocked out in the brain, not the other tissue. So yeah, the next step is to generate a liver-specific insulin receptor mice, and that's what we're in the process of trying to do and understand how that might play a role not only in high-fat diets, but other fatty liver conditions and obesity. It really increases the risk of liver disease, cancer, and insulin signaling and growth factor signaling plays a role in cancer. So we're hoping there might be a link within that.
Troy, how do you compensate for the lack of IGF-1 signalling when you knock out the insulin receptors? Because certainly in the osteoblast, the insulin receptor is mainly targeted by IGF-1.
Maybe in the osteoblast, but we've got a separate IGF-1 receptor in tissues, which is a lot more sensitive to IGF-1 than to insulin. So that's the only way really telling them apart is how much more sensitive. So we see.
Do you measure the IGF-1 levels?
we measure. We didn't measure the IGF-1. I can't remember, but we definitely measured the IGF-1 receptor levels. And
I saw it at one stage.
So phosphorylation is always both because you can't differentiate between. But I don't think I have it there. But the IGF-1 level, receptor levels were changed.
Yeah.
But that could be another reason that we don't see much of a lifespan effect because most models target both. This is just for the insulin receptor, this knockout model, so the IGF-1 wasn't affected.
Troy, this is really not science in it, but you comment on the fact that in terms of the input-output measures that 40 to 70% of it's genetic.
Yep.
And yet, what's striking to me is when I see pictures of people from 80 years ago on a farm, huge protein fat diet.
Yep.
Skinny as hell.
Yep.
So something's changed in that process, which is not simply genetic, and I'm sure it seems to me that the supply of food is actually almost being designed to inhibit normal satiety mechanisms. Is the work being done on actually? I think we can't assume that. We can't assume the food industry will change those approaches unless government interacts. But presumably scientists can actually look at what is encouraging people with that diet intake and maybe operate on that as well.
There's lots of work going on that front, the GPL and the satiety hormones. So the kind of way I look at it is exactly as you do, but in the reverse. We know the diet has changed for most people around the world, but not only 40% are obese too. So there's got to be some genetic, and there's an interaction. There's no doubt that high-fat diets, the change in food consumption has changed it, but the genetics kind of load the gun, and the environment pulls the trigger, so you can't have one without the other.
Sure.
Basically, but on that, there is more and more coming to light, particularly around the FDO gene and variants. That is really quite common. I mean, it's 2 in 6 or something. 1 in 3 people have these comments that change how they respond to hormones, ghrelin particularly. So now we've got this diet, and that's going to be very hard to change. And people are being told all the time what to eat and what not to eat, and to exercise more, but it's not working. I think we need to come to more targeted approaches, as in understanding the mechanisms for how these genes might regulate. And look, if we come genetically test them, like you've got the FDO variant, we know you get hungry more with this food, you stay away from this food. Actually, you respond really much better to exercise than this person here. This is what you need to do, and rather than a global approach for everyone. And that roundabout way addresses your question, or?
Yeah, no, it wasn't a very clean answer anyway. Just interesting. How those loops, we tend not to consider the entire, because we don't consider the whole loop round.
I think it's becoming more and more evident that the role of the basic biology behind it too, particularly with things like the Krebar variant has just been sort of found the last couple of years in Pacific Islands, Samoa, so 60% of people have this variant, and this increases BMI by huge twofold, so more than 2 to 3-fold that we see with the second most prevalent one is FDO. But these guys are actually protected from type 2 diabetes also. So just because they've got a bigger BMI doesn't mean. So there's a lot going on. And Lindsay's work showing that people of Pacific Island and Maori descent, we class BMI, being obese as BMI 30. Well, Maori and Pacific for the same percent of body fat, they're about 34, BMI of 34. So the classification's wrong.
The bones are better. But to answer your question about the farmers, I mean, not answer your question, but there's a lot more farmers with osteoporosis now than there ever used to be. And that's 70%, the genetically 70% dependent too. But it's the quad bikes and the thing out, they're just not exercising as much.
No,
but it's incredible the increase in osteoporosis.
They used to die of cardiovascular disease though.
The question at the back.
Yeah. Well, I'm not starting talking about cover. I'm not medical, I'm not an expert on this, but have you considered using euglycemic clamp, or what's called glucose clamp and insulin clamping, instead of glucose tolerance test and insulin tolerance
Yes,
test?
most of these ones we have clamped as well. These
And
are
using the technique from 1978.
Yeah, it's not such an easy technique though. There's a few people that were soft up here is on this list because we clamped with him, so it's quite difficult those sections. We hope to get that set up here, but we have clamped all these mice and we see. So the clamp basically tells you not only how the glucose is being used, but where it's going. There's more going into the muscle, is there more being released from the liver, and it's a bit more of a technical. And the mice are usually done after you've done the clamp, so that's why I presented the ITT and GTT data rather than the clamps.
ICC and data just have presented the
And mice, it's easier in humans.
to do it.
All right, so let's thank Troy again. Please join us for some snacks.